Engineering at State Permittu, and this area of expertise includes biotechnology, advanced anti-cancer drug vaccine design and development, database development, computational biology, immunopharmacology, etc. And basically, he's a chemical engineer from a VIT Vellore and finished his PG in biopharmaceutical technology from ACT Anna University and completed his PhD in biotechnology from Anna University in 2020 and Sir has obtained three patents and he has guided a number of students which includes 55 undergraduate students, 9 postgraduate students and he has acted as a doctoral committee members for 5 research scholars and currently is guiding the 3 PhD scholars and he has published a number of journals in the Scopus indexed articles in the number of articles in the Scopus indexed journals and he has organized 8 faculty development programs, conferences and seminars and he has been invited by several institutes and companies to deliver guest lecture in different areas of molecular interaction and he has received a number of awards which includes Young Scientist Award and Bharat Excellence Award and Sar is here with us to deliver a case study which he has done with the help of this Discovery Studio on molecular interaction. I request Dr. Praveen Kumar to handle this session, please.
Thank you very much, Dr. Sujata Lagnathan, madam, and the HOD, sir, Dr. Devendran, sir, to provide me an opportunity to take on present topic. Is my voice audible to everyone?
Yes, sir. It's audible, sir.
Glad to interact with all the participants of the national workshop on bioinformatics with special focus on molecular interaction. So just going to take a topic on molecular interaction. So generally in bioinformatics, we call this as molecular talking. So I will be discussing various case studies from the publications made recently. So highlight. So majorly we have to know about what are the different, what is computer-aided drug design and what are the methods. And we also discuss case studies like drug discovery hackathon competition. And I take much computer-aided drug design on treatment of cancer as well as in development of antiviral drugs. And also discuss about the database developed in my institution and projects, the funded projects happening in my institution, and the patents related to cancer, and what are the various certificate courses I'm handling with respect to bioinformatics. Sponsored projects and research consultancies for past 3 years. So if we see, so can I switch off my video alone? So may you have, I think, yeah, now I take the presentation. Yeah, so the sponsored projects like I'm working on a DST funded project in IIT Madras in Protein Bioinformatics Lab under the guidance of Professor M. Michael Komiha, who is India's #1 bioinformatician. So in his lab, Protein Bioinformatics Lab, he has developed more than 20 tools and 20 databases for molecular interactions, molecular interactions, and each and every database in a tool is a product of a PhD scholar. So he has developed plenty of tools for the benefit of society, bioinformatics society, and also guide projects in various private engineering colleges, and also received grants from Tamil Nadu government for a UG projects, and even the scholars who are working PhD under my guidance, they are receiving a research fellowship. Okay, so they are receiving around this fellowship. And presently, I'm also working on another funded project. So this funded project financial grant sanctioned for financial year 24 to 25. I received 5 lakhs out of maximum 10 lakhs because the similar part of a project I already received a DST grant. Only 135 innovation circuit grants out of 1 lakh 5,000 submissions. So this is a phase 3 pitching event interaction with Assistant Innovation Director, Sri Dipan Sahu. And get inside our topic, how researchers discover the drug for a disease. Okay, so that as a scientist, we should know we can use the computational studies to discover drug. So, what is drug design? Generally, drug design is referred as a rational drug design, or we generally try to find the drugs from a biological source. It can be marine sources. It can try from the herbal plants. We can try with animals. So, whatever we try, if that can able to cure the disease, we use it for treatment, isn't it? So, this kind of a computer the drug design can be of two types. One is traditional drug design. So earlier, 50 years before, we used just the pestle and mortar, whatever plants, whatever tissues we extract from the animal or extracted from the marine sources, we use it as a drug. So we get a combination of the drugs. We are not sure what drug is going to exactly cure from the components what we have extracted. So if we have extracted the exact drug, then we can reduce the dose of drug, and we can also reduce the number of dosages of a drug. So, which is most important, isn't it? So, instead of going with the traditional drug design, that is taking multiple compounds and treatment for disease, that is initially starting with preclinical studies and then going with four clinical studies, we can better go with a modified kind of an approach, that is structure-based drug design. In structure-based drug design, uh, in in structure-based drug design, one has to know the 3D structure of target, that is a disease receptor, and one should know the 3D structure of the ligand, that is a drug or inhibitor. If and if you have a knowledge on a 3D structure of a drug and a 3D structure of a disease protein, then one can able to find the binding affinity of a drug with a receptor, that is ligand with the target receptor, and then we can come across all these potential benefits, that is reducing the dosage and reducing the number of dosages. Okay, so this is what we're recently using, the structure-based drug design. And if you see this drug discovery in areas of modeling and simulation, we can find various areas. Generally, if you see this picture, now this picture I've extracted some 10 years before, a decade before. So for computational studies, almost we have to take nearly 3 years. Okay, but now the computational studies are completing within 1 to 2 months. You know how I am telling, even the computational studies are sped up to complete within 1 to 2 months time. So when we encountered the pandemic situation, that is when we found SARS-CoV-2 infection occurred in India and all around the world. Okay, whatever drugs present in databases. Okay, like if you see Chink 15 database, where we can find nearly 1.3 billion compounds. 1.3 billion compounds are available in one database. That 1.3 billion compounds, they have used a virtual screening method to find which is the best lead compound for spike glycoprotein of SARS-CoV-2. So spike glycoprotein is, I think, spike glycoprotein is the important target for SARS-CoV-2. And for the 1.3 billion compounds are docked with that spike glycoprotein, and they came up with the results within a span of 1 to 2 months. So because we have supercomputer, okay, supercomputing facilities, especially a California-based research scientist, research professor has come up with these kind of a results. It means that whatever studies we can able to make for emerging infectious diseases, for even a new kind of mutation in a cancer, so one can able to complete the computational studies within a span of 3 months. Okay, so after computational kind of computer-aided drug design, one can go with the preclinical development and clinical trials for checking the efficacy of the drug. Okay, if the drug fails in any of the trials, then obviously we have to reject the NDA, that is new drug investigation, whatever we make, and we have to go with another kind of a drug molecule. Okay, so and if you see this, we find so many computational areas like metabolism modeling, manufacturing modeling, protein structure prediction, homology discovery, pharmacokinetic modeling, toxicity prediction. So these are various computational areas we can able to apply in computative drug design. So, how to revolutionize the drug discovery in silico? How a drug is found for a particular disease, that will be exploring. See, if you see here, this green color, this color molecule is a drug, and the disease protein receptor you can find like this in the software, isn't it? So, a drug is binding with the receptor here. Okay, so the receptor is shown in secondary structure of a protein. Secondary structure, you can find this protein is having more alpha helices. Now let us discuss about the various methods of drug design. So these are the various methods. Initially, if you want to go for drug likeness prediction, mole soft, mole inspiration, ADMET prediction from these tools, and ADMET is absorption, distribution, metabolism, excretion, toxicity. In addition to the ADMET, we can also include L meant for liberation. So whatever tools I've showed, so most of the tools are freewares. So free open source software, one can able to work it in online itself without downloading. Okay, so we can, if only if you have a 3D structure of a ligand and a receptor, you can work with all these computer-aided drug design software, except few software. Except few software that is whatever mentioned like PS. Okay, this is a commercial software. Discovery Studio Ligand Fit. And we can find DSQS. This is also a commercial one. Okay, but the other softwares, most of the softwares are freeware. Okay, so now let me go to the next slide. Drug likeness prediction. So how the drug likeness is predicted? So the drug likeness is predicted from numerous filters. The popular filter is the Pfizer rule. So according to the Pfizer rule of five, okay, the molecular weight should be less than 500. This is first property. And whatever chemical compound you take, which you can tell as a drug, okay, if you want to term a chemical compound as a drug, so it has to satisfy Lipinski rule of five. You know, there are totally four filters. There are four properties in Lipinski rule of five. One is molecular weight. The molecular weight of a compound should be less than 500 gram per mole. The second parameter is the law of partition coefficient, that should be less than 5. And the third parameter is hydrogen bond acceptor should be less than 10, and hydrogen bond donor should be less than 5. So these are the four properties of Pfizer rules of 5. So if it satisfies any three out of four, then we can tell it is the compound is behaving like a drug. This is such a kind of drug likeness filter, okay, which uses four principles. But there are also other drug likeness filters like Weber. Okay, Weber uses the original filter like rotational bonds. The rotational bond should be less than 10. And even there is a slight variation with respect to these properties. The molecular weight should be in between 450, like 350 to 450. Partition coefficient should be 2.5 to 4.5. Okay, there's slight change in properties will be there. You got it. So there are different drug likeness filters. Especially we use this method to find whether a compound behave like a drug or not. Okay, so whatever rules they apply, we have to apply it, and we have to check. It can be the compounds can be generated from plants, or even the compounds can be generated from marine sources or from animal tissues, whatever we generate. So whatever metabolites, whatever we take, okay, first we have to check this ADMET properties. I mean, we have to check the physicochemical properties of a drug through drug likeness prediction. So we can also use this softwares like MolSoft, Mol Inspiration, and Swiss ADME for predicting the physicochemical properties of the drug. So one such output derived from MolSoft. So if you find in this graph, okay, so what if our drug is lying between minus 1.5 to plus 1.5, then we can tell that your compound behaves like a drug. So just because the drug likeness score is minus 0.82, which is lying between minus 1.5 to plus 1.5, so you can tell. So in addition to the properties like molecular weight, hydrogen bond acceptor, tone, or partition coefficient, we also take this model score in consideration in MolSoft software. ADMET. This is another property. Next to the drug likeness prediction, one can go with the ADMET property prediction. I told you already what is LADME. The T stands for toxicity. So this is important pharmacokinetic properties. If you want to check the pharmacokinetic properties of a drug, one can able to check all these properties through the softwares, the same softwares, with ADME, QuickProc, Straw Drop, MOE, KNIME Notes, PSSS Talks, etc. Then we can come across an output from this ADME. So an output we can where we can find not only the drug likeness prediction, but we can also find ADME properties. Like A stands for absorption, gastrointestinal absorption. If you are taking a drug in oral route, the absorption should be high. Yes, it is high. If it is desired, you can check whether if you want a low, then you have to check whether you're getting low or not. If you want to absorb a drug more in the gastrointestinal tract, yeah, then you can tell this high is the preferred property. And the next to the absorption is distribution. The distribution we check from the blood-brain barrier permeability. Distribution is whether your drug is distributing appropriately to the tissues or not. Like if you're targeting liver cancer, your drug should target maximum of liver. Okay, it should not penetrate to the brain. The liver is actually a kind of still hydrophilic nature, hydrophilic membrane. But if you see, brain is more lipophilic. Even though you have a lipophilic membrane in the liver, it is less lipophilic when compared to the brain. The brain is more lipophilic in nature. So if your drug is also penetrating the brain, okay, then it can lead to the hazardous effects. It can lead ill effects. Okay, unless you're targeting a neurological diseases, okay, the drug should not be a more lipophilic in nature. Again, I'm telling you, the more lipophilic a drug in nature is more lipophilic in targeting. Okay, if the brain membrane is more lipophilic, so whatever drug you treat for cancer, it can be a liver cancer or pancreatic cancer, it should not target the brain. Okay, for that you will be checking the property of a drug. If it shows no, that is a desire. Okay, unless you go for a neurological disorders treatment like absorption, distribution, metabolism, excretion. Okay, all these things we can check with the parameters. Metabolism check with the cytochrome P450 enzymes, and excretion is check with the skin permeation. So I told you the different filters, the Lipinski, Goose, Weber. These are some of the filters we can find. And pre-admit tells you also about the drug likeness prediction, toxicity prediction, ADME prediction, etc. QSR stands for the quantitative structure activity relationship. And the like quantitative structure activity relationship is generally a ligand based approach. A ligand based approach, like if you have a properties of some 10 ligands, and if you don't know the property of the 11th ligand, okay, so if you want to know the property of the 11th ligand property can be determined. The activity of the 11th compound can be determined from the already existing 10 compounds. So this we are able to make it up using developing a training data set, and whatever you're going to check, that we consider as a test model. So through the test model and from the training data set, you can get the unknown activity of the compound. Okay, like how you determine an activity, even property can be determined through the quantitative structure property relationships. And similarly, reactivity can be determined through quantitative structure reactivity relationships. And if you have more similar compounds in a plant, okay, if you want to distinguish the compounds, you can use chromatographic relationships. And for toxicity, you can use QSDRs. And for electrochemistry, you can go for QSER. And for biodegradability, one can go with QSBRs. Now go with another method of the computer-aided drug design, that is pharmacophore mapping. So this is a hybrid-based approach. This uses both ligand structure as well as the receptor structure. Both are important, of course. Okay, for pharmacophore mapping, majorly we use the ligand. So this is a majorly it comes on ligand-based approach where the functional groups of the ligand is majorly determining the recognition of binding with the binding sites of the biological macromolecules. So if you have a functional group like OH, okay, so which is more desired to get a binding with the receptor molecules, okay, OH molecules. So one should know how many hydroxyl molecules you have, how many alkyl molecules you have, how many amine molecules you have. Okay, so that's most important. Where the pharmacophore mapping majorly focus on the molecular features of the ligand, which gives you an idea about whether it is going to bind with the binding sites or not. Okay, so this is before docking, one can able to try with pharmacophore mapping or modeling. The best example of the software what we can use is the Jing format, the freeware, which we can use it for pharmacophore modeling. This is an example shown for a compound called pacillin. And okay, okay, and we can go with the molecular modeling. There are different methods of modeling of proteins. One is homology modeling, the other is threading, the third one is the ab initio protein modeling. Homology modeling, threading, ab initio protein modeling. For homology modeling, one can try with this model, 5D2, Yasara, etc. If you want to try with ab initio, one can try with these softwares. Now let me go to the molecular modeling validation. Even though we predict the structures of the models, or I mean protein models, one has to validate the stereochemical quality of a protein to assess the quality of a protein. Unless the quality of a protein uh, is good, we can't able to take this protein for docking and simulation purposes. So Ramachandran plot is used for assessing the stereochemical quality, and the number of favorable residues should be 80 and above. Okay, if you find 80% and above, then that is favorable residues. If you find in Ramachandran plot, then it can take the predicted protein structures from computational methods for further docking and simulation analysis. Molecular docking. So, what is molecular docking? Molecular docking, that's what we call it a molecular recognition in various areas of biology. So, if you see in various areas of biology, like if you see this as an example, antigen binding with the antibody, where we can find this an example in immunology, isn't it? In immunology, majorly, we discuss on antigen antibody interactions, where we can find 99% or more affinity, that is an antigen, whatever we use, it should more bind with the antibody. Generally, the antigen is from a disease protein source. If it is a viral, if you want to treat a viral diseases and if you want to use an antibody, then you take a viral antigens and you synthesize antibodies for that viral antigens. Okay, so the antibodies can also specifically target the antigens in eliminating the aggressiveness of the disease. And similarly, if you see the second area of biology, enzymology, where you can find enzyme bind with a substrate. You remember, if enzyme bind with a substrate, you can find the enzyme substrate intermediate complex, and after the reaction, you can find enzyme and product is released, isn't it? So, if you want to get an enzyme followed by a product, the enzyme substrate binding is essential. So one should know this molecular recognition. And similarly, if you see this third thing, that is signal transduction. The example what I can provide transport molecule is the hemoglobin. You know, the hemoglobin generally carries oxygen during inspiration, and exhalation it's going to eliminate CO2. So the hemoglobin, when it takes the oxygen, it forms oxyhemoglobin. So that is a different protein. And if you try to exhale, leave out carbon dioxide, then you can find the protein molecule is getting as in the form of carboxyhemoglobin. So what you understand, oxygen and carbon dioxide is obviously carried out by the hemoglobin, and this kind of a signal transduction is also a kind of a molecular interaction and a signal receptor. For this, I can give examples like again immunology. If you see IL-4 ligand binds with IL-4 receptor, IL-2 ligand binds with IL-2 receptor. GM-CSF ligand binds with the GM-CSF receptor. So you can find many immune molecules, okay, the cytokines bind with the corresponding cytokine receptors. So whatever examples I provided in different areas of biology, okay, we can able to study everything through a computational technique called molecular docking, where in molecular docking, we just not only study the affinity of your ligand binding with the receptor, but we also able to determine how well a binding affinity can be measured through binding energy. We determine binding energy Through the binding energy, we distinguish which ligand is binding well with the target receptors. Okay, so this is something about molecular docking. In molecular docking, we have two major steps. One is a search algorithm, the other is the scoring function. So you know that there are three ways like autodock to dock with ligand with a receptor. Okay, so whatever I tried. Okay, whatever I tried with the publications can be from low impact factor to the high impact factor. I tried one day with the autodock. But if you have a commercial software like Discovery Studio, I advise you to use that commercial software. The reason behind why I'm asking you to use the Discovery Studio is here in Discovery Studio Ligand Fit, okay, it's going to use 8 scoring functions. The Autotalk is going to use only one scoring function, and Discovery Studio Ligand Fit is going to use 8 scoring function. Like based on these 8 functions, we can able to analyze, assess the quality of binding. Okay, so the most important thing is what search algorithm they have used in determining the binding sites and determining the binding affinity, determining the hydrogen bond interactions, van der Waal interactions. Okay, what algorithms they have used. Okay, and other than that, what scoring functions they have used. For scoring function, even in Discovery Studio, we have a module called the DS ligand score. The DS ligand score is going to determine 8 different scores. And then what is lip dock? So the difference between ligand fit and lip dock is in ligand fit you can dock a single ligand with a single receptor. But in lip dock you can dock multiple ligands with a receptor. Like if you want to screen a multiple drugs with a single target, one can try with lip dock module present in Discovery Studio. Okay, similarly we have a commercial software called Glide, which is a docking software present in Scroidinger package. Scroidinger package, Discovery Studio is a package. Inside you can find so many softwares. Similarly, for molecular docking of a ligand with a receptor, we can generally go with Glide. Okay, a small molecule binding with a receptor. If you want to dock a macromolecule with a macromolecule, okay, that is protein versus protein docking, you can use Z-dock. Z-dock is a software we use for docking. In Scroidinger, we have Gold. In Discovery Studio, I have Z-dock, but in Scroidinger, we have a tool called Gold, which docks a protein versus protein and protein. Got it. So the softwares vary whether you are docking a small molecule with a receptor or a protein with a protein. Okay. So this is something about docking. Majorly, you have to remember is one is the search algorithm and scoring function variates the freeware with the commercial software. And this is one such algorithm used in Discovery Studio Ligand Fit to determining the binding sites. You know, generally a protein molecule binding sites are determined by putting the protein molecule in a grid box. And whatever fluid, we generally use water for filling to identify the hydrophilic binding sites and hydrophobic binding sites. So whatever water molecules present on the outer surface of the protein, we call that as an hydrophilic binding sites. And generally, the buried sites of the protein is what we call it as a hydrophobic sites, also called as lipophilic. So generally, if fill the water molecule, water as a medium, you use it in the grid to determine the hydrophilic and hydrophobic binding sites. And docking. So if you see docking, okay, most of the molecules dock with all the receptors, but the efficient docking we can determine only through the number of hydrogen bond interactions. So if you see the hydrogen bond interactions between an alkyl hydrogen bond acceptor and an alkyl hydrogen bond donor, you find only one. Here in this case, you find only one hydrogen bond interaction. But if you see a ligand which is having an aromatic ring or nearly 5 membered ring, and even you have a tyrosine amino acid in a protein which is having a 5 membered ring, and if you can able to find 5 hydrogen bonds, that is 5 hydrogen bonds from a ligand is interacting with the 5 hydrogen bonds with the receptor. Okay, so you can find more hydrogen bonding, so more energy you can derive after the docking. So here in this case, you get a less docking score. But here in this case, if you find this kind of interaction, you find more docking score. That is in autodock, we expect actually negative maximum binding energy. And in discovery studio, we expect maximum score. Okay, so maximum score or negative binding energy from autodock we can get only through the hydrogen bond interactions. So in this case, aromatic interaction shows a best interaction when compared to this. An electrostatic can find this kind of interactions, two interactions. Hydrophobic it can end with four interactions. So there are different interactions, not only the hydrogen bond interactions, we also have other interactions like electrostatic and hydrophobic. Also, it can mention you like how much effectively a ligand is binding with the receptor. If you see a ligand is binding in different poses. This is a stable pose, but you also find the other poses in animation, isn't it? So how effectively a ligand is binding with the receptor, one can able to check it even through simulation. Okay, even through simulation, you can check. Okay, and here also you can find how effectively a ligand can bind with the receptor. In hydrophilic binding sites, you find very less hydrogen bond interaction. But if it is buried inside, that is, if you find your ligand is binding in the hydrophobic binding site amino acids, you find more interaction. So this is most preferred. When compared to hydrophilic, hydrophobic interaction is more preferred. Like bonding, we have types of interactions, bonding interactions and non-bonding interactions. Generally the bonding interaction is the sum of bond stretch, this is bond stretch, and this is bond bend, and this is rotate along bond. The sum of energies of all these three types of interactions is what we call it as bonded interaction energy. U stand for the energy. The bonded interaction energy is determined through these three energies. So, like the summation of the non-bonded energies is what we calculate. That is the sum of van der Waals and electrostatic we calculate as non-bonded energy. And van der Waals energy formula is represented by this, and electrostatic energy is given through this. And the sum of bonded and non-bonded is what you calculate for total energy. So whatever we use concept for molecular docking, the same concepts we can also use it in molecular dynamic simulation. Then what is the difference between docking and simulation? So in docking, we generally check how effectively a drug is binding with the receptor. But in simulation, you check also with respect to time. In addition to the docking of ligand with the receptor, how effectively this interaction is there with respect to time. That you can study through molecular dynamics simulation. Okay, so obviously, whatever you use the boundary interaction, on-boundary interaction formula, you also find the same in molecular dynamics simulation. And the softwares we generally use is the Promax. In IIT Madras, I'm using the Promax for doing the MD simulation. Other than that, we can also use Amber, NAFT, and Desmos. Now, we can see an example like if these are some of the computer-aided drug design methods. Okay, we can also say pharmacokinetic, pharmacodynamic modeling of cancer. We can able to try if you want to get a precision medicine for a cancer or a specific disease. Okay, we can try with system biology tools. So there is one tool called Symbiology from MATLAB. So in our institution, we have a Symbiology license for each and every faculty and a student. Okay, one can use this as a MATLAB online or offline software for pharmacokinetic modeling. If you want to develop compartment models, single compartment model, two compartment, three compartment models, one can develop the models and we can give the values for the kinetics, okay, and, for the rate constants, and one can able to determine the drug dosage in each and every compartment's passage. Okay, so one can know how much drug is utilized and how much is excreted. So through the system biology tool present in MATLAB software. Now we can come across the other method of computer-aided drug design, that is virtual screening methods. So generally, we are not focusing on a single drug, isn't it? So earlier, to tackle the pandemic situation, that is, while tackling the discovery of drug for SARS-CoV-2, so what we have done, we have taken the clinicians have generally done by treat. They didn't treat actually, but they didn't treat properly because they have used only repurposing drugs for treatment. So for treating SARS-CoV-2 in 2019 to 20, many clinicians prescribed 8 types of drugs. The one drug is for anti-dengue, one is anti-malaria. So they also used anti-HIV. How come an HIV drug can able to treat the SARS-CoV-2 infection? But the clinicians don't have any idea about how the disease can be treated. So they have used all the kinds of a drug. But exactly if you want to treat a particular disease, okay, one has to go to use the multiple drugs. Okay, one has to screen the drugs with the multiple drugs used for treating viral diseases using virtual screening methods. This also is for many applications. I'm telling an example for treatment of infectious disease. So flowchart of virtual screening. So virtual screening can also be a ligand based or a receptor based. Structure based here I mean a receptor. That is here you want both the ligand and receptor 3D structure. If only if you have, then we can go for docking and we can tell which is the best from the docking results. But this is time consuming. Okay, instead of going with the normal docking, one can go with virtual screening docking software. And we also try with ligand-based methods, which is rapid. So one can go if the actives are known, go for similarity searching and pharmacophore mapping. If inactives are known, that is, if it is a newly synthesized drug and not yet tried with any kind of research studies, you can use the machine learning methods. You can use the machine learning methods. Okay, so these are the types of virtual screening methods. Either you can go for ligand-based, or you can go for structure-based, or we can go for hybrid-based approaches. Okay. So what are the different virtual screening methods available? The first method what we'll be discussing is lead hopping. In lead hopping, see this is the initial lead identified. If this lead compound is identified, if this compound is marketed for treatment, but if this is not precisely targeting the patients, that is only 1 in 4 patients are getting treated or 1 in 3 patients are getting treated, and if you want to improve the drug, okay, one can go with lead hopping. In lead hopping, we dissect, we fragment the molecule. We fragment the existing drug and we attach with the desired functional groups. Earlier, we had this kind of a functional group, but now we had a different kind of functional group. Okay, may this functional group, whatever present, can give a better efficacy of binding with the target receptor. Okay, if you want to target specifically, one can able to fragment the molecules and can attach through the linkers and use that compound as a new lead for the treatment of diseases. So this concept is called lead hopping. There is another method called increasing high throughput screening rates. If you have a combination of peptides and you want to check which is the best one, okay, one can dock all the peptides with a target and we screen which is the best. So that we call it as a high throughput screening. Okay. And if you see the virtual screening, generally, what you infer, the generally the molecular docking is to dock a single ligand with a receptor. But virtual screening, if you have multiple drugs, if you want to screen which is the best one, you can dock all the ligands with the receptor, and we can come to know which is the best drug identified. Okay, so this is what we call it as a virtual screening. And if you have inactives, if you don't have known drugs, if something new drugs are discovered, okay, if you want to find which is the potential inactive for, which is the potential inactive for the treatment of diseases, one can also use R programming. So R programming, we have different libraries. So each and every package imported in the library has different functions, the predefined functions, okay, to develop a cheminformatics kind of a software. So whatever you try with the cheminformatics software, you can able to work with your own created program. So it means, I mean that using R programming, you itself can create your own software, okay, for screening the molecules. One can develop the software using R programming for screening the molecules. Now, let me go to the strategies of computer-aided drug design for anti-cancer drug. So, if you recap whatever we have discussed, okay, initially we have seen structure-based approach like molecular docking, which uses both the 3D structure of a ligand and receptor, and molecular dynamic simulation, pharmacophore modeling, pharmacokinetics modeling. This is through MATLAB. Fragment-based drug screening, you can use commercial softwares for fragment-based. Quantitative structure, you can use again Discovery Studio. Okay, and ligand-based approach, one can go with uh, uh, QACR. Okay, now if you see these compounds, there are different compounds, and based on the inhibit properties, we can able to target. This is an example like oxindole conjugates can target CDK4 enzyme activity. So the anti-cancer properties I can find is cell cycle arrest and apoptosis. Okay, general interacting residues identified out there. Similarly, carbohydrate-based compounds can uh, can able to target the serine and threonine kinase receptors. So this we call it as an inhibitor. So like this, even if you see the cancer drugs are discovered based on the targets, and even the binding site amino acids are determined are also available in various biological databases. Okay, now let us come to the case studies. So till now we have discussed about various methods of computer-aided drug design. Now let us discuss about the case study one. So this is a hackathon competition organized by ASD, CSCR, a Ministry of Education, Government of India. So in 2020, during the pandemic time, they have established 30 problem statements. So this kind of a competition is open to all the faculty members and students. So anyone can able to register and can try with it. Okay. So attractive uh, prize amount was there. So I have guided a team of 6 members. So these 6 members have won 3.6 lakhs in the competition. Like a Smart India Hackathon competition, this is one such competition, Participate Hackathon, hosted by Government of India. So I have taken this as a problem statement, identifying covalent spike protein inhibitors. Okay, not just spike protein, covalent spike protein inhibitors to control the infection of SARS-CoV-2 from small molecule databases. I didn't utilize even commercial softwares, even though they provided many commercial softwares for use. So for participating, they have provided many commercial software so what we can work in online for four months during the competition. But we have used the freewares alone to get this kind of a grant, lump sum 3.6 lakhs grant. Actually, well, if you want to go to this kind of a research, one should know the important target receptors of SARS-CoV-2. One should understand the genome of SARS-CoV-2. So if you see the SARS-CoV-2, nearly 4 structural proteins are there and 16 non-structural proteins are there. The most important protein is a structural protein, specifically a spike glycoprotein. If you see the open and closed state of spike glycoprotein, okay, we have the spike glycoprotein, we have different conformations, different structures. We can have a closed state spike glycoprotein, open state glycoprotein. Okay, so not only we have open and closed, but we also have semi-open spike glycoprotein. So what do you mean by that? What is the difference between closed, open, and semi-open? So actually, if you see the spike glycoprotein is the protein present in SARS-CoV-2, generally binds with AC2 of a human. It generally binds with the AC2. Okay, angiotensin converting enzyme 2. So spike glycoprotein interacts with the AC2 of humans. So whatever it binds, okay, the binding sites of the AC2 on the spike glycoprotein, that we call it as a receptor binding domain. That we call it as a receptor binding domain. Okay, instead of going with a closed and open, if you try with a semi-open, you may get a precise medicine. You may get a precise drug for that. So one should try with both closed and open, and also try with semi-open, whether you are getting a best binding energy or not. Okay, so if you can able to target the binding sites of AC2 on the spike glycoprotein, then it can tell that is the best drug, that is the best inhibitor. Okay, so for this, I just tried nearly 13 binding site domains. 13 important binding sites are identified in the receptor binding domain. Uh, so these are the 13 binding site amino acids. So in the 13 binding site amino acids, whatever we identified in the region 319 to 541 amino acids of 1333 amino acids by glycoprotein. Okay, so I can able to find uh, I have to find the best drug. Okay, so this is my objective. So I have taken 1151 compounds from PubChem. So to find the best drug for the semi-open spike glycoprotein, I first tried with downloading the structures of chemical compounds from NCPA PubChem. So in NCPA PubChem, I found 1152 in 2020. Now I think we get almost 1700 compounds in NCPA PubChem, which these drugs are generally used for treatment of SARS-CoV-2. So at that time, I tried this much molecules. Initially tried with virtual screening through Autodoc PYRX. Okay, I identified 100 base compounds out of 1152. Okay, and then I came up with the screening of 20 base compounds. The 20 base compounds are screened using the Autodoc software. Okay, and from the first 100 base compounds I identified through ADME, Swiss ADME, and from the 100 base compounds, I want to identify 20 base compounds. This I have done through Pyrex. Okay, and from the 20, if I want to get the best 10, this I have done with the Autodoc. Okay, so this is a pipeline to show how to how to work on a project, how to screen the best drug for a specific target. So we got 10 base compounds, and this I checked for hydrogen bond interactions, number of hydrogen bond interactions, and the exact binding sites. Okay, so from that I identified the top three base compounds, and from the top three base compounds, the first base compound what I identified is the ethafrin A. Ethafrin A is the steroidal lactone compound present in ashwagandha or Indian ginseng. Okay, so this is the compound I got from the NCBI PubChem database, which is targeting specifically the spike glycoprotein. So not only I found four binding sites out of 13 binding sites, but also I can find in MD simulation, it is showing a good stability after some time. And similarly, even we find less fluctuations. Okay, when I talk the vitafrin with respect to spike glycoprotein. And the case study two is to identify a best vaccine component for Nipah virus infection. You know, the SARS-CoV-2 emerged in 2019, declared as an pandemic in March 2019. But you know, even in India, in Kerala, in 2018, okay, a few people get infected with Nipah virus. You know, there is no treatment for Nipah virus. So only they have to isolate the people and they have to sacrifice. That's what happened. Okay, so just because the treatment can't be given. If so, first one has to, even if you want to treat such a kind of infections like Nipah virus, even though it's not showing significant mutations, even a single strain targeting is more difficult. So this can be achieved. For this also, we can able to bring a vaccine. So through reverse vaccinology approach, this is another competitive drug design. We call this as immunoinformatics approach. Okay, so either we can go for T cell epitope based vaccine design, or you can also go with B cell epitope based vaccine design. So this paper got published in 2020 and got multiple grants. Even we have selected for INAE National Award, and even we got a project grant from Government of Tamil Nadu and awarded as a best project. Okay, so this is a computational technique I have used, and we have used Nipah virus W protein as a target. So here, this is the immunoinformatics approach we made. Okay, so we predicted a T cell epitopes through various tools, through various immunoinformatics tools, and we've identified the best epitope for MSC1 and MSC2. Okay, one best epitope for MSC1 and one best epitope for MSC2. So whatever I determined for MSC1 initially, I tried talking with Vitafrin. I tried talking with the epitope, okay, the peptide, with the peptide, what I screened in immunoinformatics. And similarly, whatever peptide I identified as the best, I tried talking with the HLA, that is MHC2 molecule. So then I whatever energies I obtained, I just tabulated. Then I have gone with MD simulation. So majorly I've used talking simulation to construct a multi-tope for treatment of Nipah virus infection. Okay, so if you see in SARS-CoV-2 in the immediate year, okay, we had a vaccine developed by we had a vaccine developed by US people, called COVAX. You know, the Covishield has given only 76% efficacy after second dose, and Covaxin has given 91% efficacy after second dose, after booster dose. But COVAX, this vaccine is giving more than 99% efficacy. It means that whatever multi-dose vaccine you discovered through immunoinformatics approach can be used for treatment of effectively used for treatment of viral infections like SARS-CoV. So if you compare COVAX vaccines with other things, okay, so you have plenty of advantages with the COVAX vaccines when compared to monoclonal as well as the small molecule. Okay, so now you can realize that even the multi-top design through reverse vaccinology is a potential approach for the treatment of infectious diseases. So in a veterinary, as a veterinarian, you may experience a lot of viral, bacterial diseases in the animals. So one can also able to go with these kind of approaches to discover an peptide-based vaccine, okay, for the treatment of the disease, other than simply treating with small molecule drugs. And this is the case study 3. Here in this study, I just tried with a same plant compound called vitaferin, which is a steroidal-acting compound derived from ashwagandha, whether is it used for treatment of cardiovascular disease or not. Because most of the drugs has a side effect it can give a it's not best for the cardiac muscles, isn't it? So many drugs, they are prone to get heart attacks. So I tried this kind of effect of this vitafrinate drug with major target receptors of cardiovascular diseases. When I tried, okay, vitafrin is shown as the best inhibitors, okay, for the receptors like PRP1 and P2Y1. So I just tried initially with 9 receptors. I generally tried talking with 9 receptors, and I also checked with standard compounds. So for PAFH, this is the standard compound used. So I tried talking with these compounds and tried with the vitafrin A compounds. So when I tried talking, I found for this PRP1, the vitafrin A has shown a significant result. And like the PRP1, I can also find what is the other which is showing? P2Y1. These two alone I can show. The Vithaferin A is showing the best result when compared to the standard ligand compounds. Similarly, DSX score is determined. So in this case study, we have used Autodoc, we have used MD simulation using GrowMax, and also the DS score calculation analysis. Okay, so this is the next slide showing you about the 3D interactions of Vitafrin A talk with the PRP1 and P2Y1. And MD simulation studies for the protein with A is shown here. Okay, MD simulation, that is RMS to graph, RMS of graph, and binding energy. And the third case study, what I tried is with a plant compound called Zoyrevinone. The Zoyrevinone is the rotinoid, okay, derived from Corevia diffusa. In Tamil, we call it as Mukaratai Kirai. So, whatever Mukaratai Kirai from that the rotinoid called Zoyrevinone B, I tried docking with the potential receptors of hepatocellular carcinoma, that is caspase 3 and EGFR. Okay, so here, what are the techniques we have used? Of course, this is also a funded project from UGC. So this is the publication. So what are the techniques we have used? We have used docking and MD simulation. So in docking here, we can find the 2D interaction images. And in simulation, we can find RMSD graph, RMS of graph, okay, and etc. Okay, so we have used docking and MD simulation for identifying uh, the affinity of BB with EGFR and caspase 3. This is case study 5. So in this case study, in addition to the docking, we also used system pharmacology approach. System pharmacology, it includes even network pharmacology. So, uh, we have utilized the system pharmacology or network pharmacology uh, uh, approach for identifying the best drug for ovarian cancer. This we published in Scopus Index Journal. So I've used uh, unraveling the pathways. We have tried initially top 1,000 genes, okay, that upregulate and downregulate the ovarian cancer. And further, we have screened with top 100 genes through p-values. And whatever 100 genes we got, like upregulated, downregulated, again, we have gone for another screening, okay, and we got the best 10 important targets. The first best 10 important targets for ovarian cancer is these are the gene names. So whatever proteins we got, the 10 best proteins, okay, I try to dock with known plant compounds, which is generally used for treatment of ovarian cancer. That is apigenin, quercetin, and resveratrol. And I want to find which is the best out of these plant compounds. When I tried docking, okay, I found quercetin is the best compound. So how I am telling quercetin is the best compound because out of 10 targets, so the quercetin is docking with a negative maximum energy. So that is if you see -6 to -8.5. So here in the second target, FGFR1, quercetin is the best targeting molecule. And similarly, the third, okay, and this is the second best interaction, third best interaction, fourth best interaction, and fifth best interaction, and sixth. Okay, this is the sixth best. So I can tell quercetin is best when compared to epigen. So what I did, again I'm telling you, I have taken 1,000 genes and I filtered to the top 10 important genes, which is connecting to the network, which causes the ovarian cancer. So when I tried talking with the plant compounds, I found quercetin is the best. How I am telling the quercetin is the best through the docking scores. The docking score is plotted LG. Okay, so I can find it's targeting six receptors out of 10 important target receptors. So I declared that quercetin is the best dock compound. And in case study 6, okay, which is published in Scientific Reports in 2022, this is used for diagnosis of first and second stage of kidney cancer. You know, the in cancer we have 4 to 5 stages. Generally, if you see BC. Participants, am I audible to you?
No, sir. Yes, sir. Now you are audible, sir.
Okay, for how much time I'm not audible? For 1 minute? Fast 1 minute or fast?
1 minute.
Okay, sir. So then I can continue. So this is the last case study I'm going to show. That is, this case study we used to identify this is just for diagnosis, not for treatment of kidney cancer. If we want to find what are the most important genes which causes first and second stage kidney cancer, because the remaining stages we can't able to treat. Okay, one has to go for biopsy. If you go for even biopsy of a small tissue, the regeneration of the tissues is difficult in terms of kidney cancer because kidney is a small organ. So one has to go for transplantation. If you go for later stages of cancer, if the cancer is advanced to the later stages, one has to go for transplantation. So only we can target the first and second. So this article is majorly focused on a computational based approach, especially a network pharmacology or system pharmacology approach to identify the best genes, best 5 or 10 genes for the first and second stage of cancer. For this, we have taken the microarray data sets. We have taken the microarray data sets. We have taken two microarray data sets, and each microarray data set We also categorize the data as stage 1 and stage 2 cancer data are available. Okay, and also we have the data are present for healthy patients as well as for the cancer patients, so that we can compare healthy and cancer patient and can find which is the potential gene, like what is the distinguished gene in the cancer patients which is more expressed, and like through that we'll be finding the best of genes. Okay, so this is the idea behind. So for this we have taken the data sets downloaded, GUI expression, gene expression omnibus data sets are downloaded from NCBI, and even the data sets are taken from the cancer databases. We have together we have taken for data processing, and we have gone for differential express genes. So these genes, so we try to find through probability distribution. Out of 1,000 genes, we find the most important 10 genes finally. But we have used various screening methods. So one important screening method is to enrich through pathways, enrich the genes through pathways. Okay, so there are different pathways. Uh, uh, there are different pathways which can result in the formation of disease, the formation of kidney cancer. Then, what are the combination of pathways which can generate kidney cancer? Okay, so that combination of pathways we have used, and we identify the genes in each and every combinations, and we cumulatively we put in Venn diagram and find the most important common genes in both the data sets, in both the microarray data sets. Okay, so this is how we have tried. And if you see this, we also use the Venn diagram for finding the genes. So the Venn diagram shows for p is less than 0.001, p is less than 0.05, and p is less than 0.01. So I have used different statistical analysis and try to find the genes which are responsible for stage 1 and stage 2 cancer. So here we also used uh, constructed a kind of network of pathways. Okay, so whatever pathways we have, we just interconnected in the pathways, and through the cross analysis and clinical relevance, the best genes or the top best genes are identified for stage 1 and stage 2 cancer. Whatever mentioned in the red, PAR, P6A, PLCV1, so these are the top base genes available. And both the publications, the ovarian cancer and kidney cancer, whatever I showed, these are published in open access. Okay, so anyone can able to go and download these articles really, and look through the treatment of ovarian cancer and diagnosis of kidney cancer. So both use a system biology or pharmacology approach to determine the important genes, and then in one article we've gone for docking, and in another article we have gone for extensive network for pathway analysis to filter the base genes for stage 1 and stage 2 kidney cancer. Okay, so these are some of the intermediate steps. Okay, like we got different pathways, combination of pathways in stage 1 and stage 2 kidney cancer. And of course, this is of course the last case study that is developed. We have developed our own indigenous database to find plant-based adjuvants for treatment of cancer and for treatment of autoimmune diseases. For this, we have used C Sharp SQL as a foreground. No, no, C Sharp and PHP in foreground. C Sharp is the we used in .NET framework. PHP is personal homepage, like how we use HTML. PHP is the server-side language, server-side web language, and SQL is used as a database language. Okay, so whatever data we generated from docking, we deposited in a database, and through SQL we fetched, and we projected the output through the PHP, through C Sharp, through .NET framework. So we have developed our own database in SVC for identifying the best adjuvants for treatment of cancer as well as the autoimmune diseases. And we also, I also published a patent in biomedical imaging system for cancer screening using artificial intelligence. Okay, and then so this is a logical logic, like whatever images we generate from the microscope or even can be from X-ray. So whatever we generate, okay, these images we can take, and through the image analysis, normalization, parallelization, the different procedures which can able to give the to the color intensity of the image and the shape and the size of the images can give you the stages of cancer. If you are checking with the stages of cancer, you can find the stages of cancer. If you are checking with the infectious source, so like what is the source for the infectious disease, you can identify the cells. So we can use it for many purposes. Here, majorly I use it for cancer detection. Okay, and there is also another second patent published in 2023 recently, that is a wireless liver cancer detector. And a summary of the funded projects. So I'm presently working on hepatocellular carcinoma, liver cancer. Okay, a DST funded project. So this is the funding amount I received, and I'm working a part of a project in IIT Madras. Recently, I received 5 lakhs from AST Government of India to work on similar kind of a project. But here I'm using a nano targeting. Previously, I'm using the mitochondrial targeting. So I'm checking how effectively my drugs are going to treat through the various drug delivery techniques. And also like whatever so in both the projects I use bioinformatics approaches. Here I used molecular docking, virtual screening, MD simulation. Here also I'm using the same. And here I have used immunoinformatics approach in the project, and I'm guiding for a private engineering, private college scholars, PhD scholars, and MS scholars. These are some of my university recognition. So I work. I'm also helping these universities. These are some of the organized events. In the last 3 years, I got funding from CSCR, DBT to conduct workshops. Even 6 months before, I received funding from AST to conduct FTP. Okay, I received around 3.5 lakhs. And these are the certificate courses approved by MSME. So I'll be usually conducting 15-day certificate course or 1 month certificate course in various areas of bioinformatics. So I've conducted in artificial intelligence and machine learning approaches of drug and vaccine design. Before 6 months, I've also conducted on system biology approaches of drug discovery in cancer. Likewise, I conduct in various specific areas. So this month I've already conducted 1 month certificate course in bioinformatics. And the next month, 11 to 25th September, I'm going to conduct 15 days internship and certificate course on bioinformatics. Okay, so this is the proposed course for 15 days. Okay, and summary. So generally, what you can infer, the structure-based and ligand-based drug design are the basic approaches we use for competitive drug discovery. But nowadays, just docking won't give you a better impact. Won't give you a better impact of drugs impact on the treatment of diseases. One should go for development of precision medicine. Precision or personalized medicine can be achieved only through establishing using the network pharmacology approaches. That is not only virtual screening can give you the best drug. You should also use identify what are the genes responsible for causing the disease. It varies from patient to patient. So one has to study the genome of a human or a animal, then precise targeting is possible through network pharmacology approach. Okay, so nowadays we are using network pharmacology approaches or artificial intelligence and machine learning approaches to solve to diagnose and treat the rare diseases. Okay, so if you want a personalized medicine, one has to use these approaches. So these are the references I have completed. Any queries, you are welcome to ask me at present. So I have completed my session. So any doubts, you can ask. Dr. Sujata, madam, are you available?
Gentleman is not here, sir.
Okay, okay. So one can inform, or if you have any doubts, you can ask me. Or if there are no doubts, I can stop recording the presentation. Any doubts? Yes or no?
No, sir.
Okay, thank you very much for your interaction. May I know about your profiles? Like, are you a faculty members from institutions?
So, no, sir, I'm a student, sir, UG student, internship student.
Okay, okay, okay. Well, see, actually, this is an extensive research. Okay, so to work on these areas, one should have a basic knowledge on what is bioinformatics, what is computer-aided drug design. Okay, so this is most important. Okay, so one should learn from the basics. I'm conducting so many certificate courses. Okay, so uh, one can able to learn all this. Okay, through the courses, and you can able to experience your research only during the practice. Okay, and I can provide my LinkedIn URL. So if you like to connect with me, you can connect with me at any time. Okay, so if you want to learn from basics and if you want to experience any projects, you are welcome. So you can contact me through this LinkedIn URL. Okay, participants, so thank you very much. Okay, have a great day to you all.
Thank you, sir.
Thank you, sir.